I, I welcome all of you and thank you for attending this event in honor of Professor Robert Furchgott, who served on the faculty of the Department of Pharmacology for half a century and who was awarded a share of the 1998 Nobel Prize in Physiology or Medicine for his discoveries on physiological functions of nitric oxide. We also are privileged to have in attendance today several members of the Furchgott family, one of whom, Dr. Furchgott's daughter Jane, will be the first to address us.
Well, it's really nice to be here today, and it's nice to be an honored guest for something I didn't do at all. But I just am really honored to be here to represent my father, who's passed away, but just to be at Downstate, where I spent many years of my life visiting and even working here one summer. And it's just like, you know, Brooklyn is dear to my heart, and it's just really great to be in this medical school again and see how it's expanded. And also how the Furchgott Foundation still exists, thanks to Rose Jackman and many other people who are really close to my dad, and that his name and his honor could be made to help the sciences, to help people here, to help students continue, and to help research, to help bring you speakers. So I'm just so glad to be here, and I thank all of you for your interest and your being here. I'd just like to say also again, it's just such fun memories. Seeing a video of my dad talking really brought tears to my eyes, but he was a really important part of my life, and none of us are scientists, but we're really glad you're here, especially the younger students who were here before too, and that you're continuing with research and taking care of people. So I'm glad to be here. Thank you.
I wish to introduce our main speaker, Professor Randall Barbour, with whom I work at the Downstate Research Incubator. He will discuss the many impacts that nitric oxide has on our daily lives and its effects on health and its role in disease, and also important spin-off developments and commercial opportunities that have resulted from our growing understanding of nitric oxide's actions. Well, originally trained as a biochemist, Professor Barbour came to Downstate to head the clinical chemistry laboratory at University Hospital. The body's mechanisms of oxygen transport and utilization play key roles in routine patient monitoring as it happens. Recognizing that importance, Dr. Barbour further appreciated that if you could perform this monitoring non-invasively using optical methods, that could be an alternative to routine blood gas determination that is completely non-invasive. So that insight has led to the introduction of a new class of technology that is called functional near-infrared spectroscopy that Professor Barbour's group has played a leading role in developing. And it has led to a new scientific field that is now widely employed worldwide. And please do come up and tell us about these things.
Where's my little guy? Oh, cool. Okay. Well, it's a real pleasure to be here, and especially it's honored to have members of the Furchgott family here. It's really quite an honor. Okay, so what I'd like to do today is just give you a broad overview of some of the understandings that have been derived over the years regarding the impact that nitric oxide has on metabolism and some aspects of clinical medicine, but also how this has affected my own research here at Downstate and prompted us to move into a completely new direction that we had not thought of even as of a few years ago. So one way to recognize the significance of fundamental discovery is to appreciate its impact on our daily lives. And experience shows that transformative understandings are often accomplished in at least two stages. The first effort is the discovery itself, which in the case of biological systems requires characterizing the principal effect and often important secondary impacts. Next, armed with these understandings, practical applications are often derived that bring benefit in the form of new pharmaceuticals, new technologies, and in some cases, new understandings yet still. A good example of this two-stage process is the subsequent understandings derived from the discovery of the structure and functional features of DNA, which has led on to understandings of the macromolecular machinery of the cell. So armed with these understandings, today we now have translated this to derive a whole field of omic technologies. There's genomics, proteomics, metabolomics, and so on. And this range of technologies, which has now produced tens of thousands of new jobs, is also considered a leading edge opportunity for the prospect of individualized medicine. So our task today is to recognize the importance and fundamental impact of Robert Furchgott's discovery that nitric oxide is an endogenously produced gaseous signaling agent that not only modulates key physiological processes that include blood pressure in the form of vasodilation, the immune response, forms of neurotransmission, and inhibiting platelet aggregation, among other factors, but exerts these effects throughout all of biology, including plants and bacteria. Additionally, our aim is to attempt to build on these teachings with the goal of introducing new technologies and understandings that I argue at least have the opportunity to have potential for transformative impact. So we begin by recognizing some of the basic properties of nitric oxide and factors that modulate its activity. Most evident is that nitric oxide is a simple molecule being composed of just two atoms, nitrogen and oxygen, and it has an unpaired electron, which means it's a free radical. Also appreciated is that it has unique chemical properties and that these properties have a large impact on its actions. For instance, it's already mentioned that it's a free radical being chemically reactive, but its reactivity is quite limited. It can only react with metal ions, such as you find in hemoglobin, or with other free radicals. Okay? It's lipid soluble. It's a very small molecule, neutral. It's being lipid soluble. It can pass freely through membranes, and it does not require a carrier. It also has a very short half-life on the order of 2 seconds, but this is much longer than what you would have for other types of free radicals, although much shorter than what you have for steroids or peptides. Also a key factor in terms of the actions of nitric oxide is to recognize the strong interdependence between nitric oxide synthesis and degradation and how this depends on oxygen availability. And no doubt driving this is the simple observation that nitric oxide and diatomic oxygen are actually very similar to each other. In fact, they're adjacent to each other in the periodic table. So you have So here we have nitrogen and oxygen, and the only difference is oxygen has an extra neutron, proton, and electron. Okay? Given that you have this similar function between electronic structure of nitric oxide, it really should come as no surprise that enzymes and carriers of oxygen in the body will react with nitric oxide and vice versa. And among these, two of them in particular warrant our attention. First is hemoglobin itself, which is responsible for binding oxygen in the lungs and transporting it to the tissues. But in doing so, it turns out that oxyhemoglobin is a principal scavenger of nitric oxide in the vascular space. So here we see that oxyhemoglobin, this is the ferrous form of iron and hemoglobin with bound oxygen, can react with nitric oxide to form nitrate, which can be eliminated by the kidneys, and the oxidized form of hemoglobin, which is also known as methemoglobin. Now, methemoglobin warrants our attention because once it's in the Fe+3 state, it can't bind oxygen, so its oxygen binding capacity is limited. And second, having undergone this transformation, the other hemes that are maybe in the reduced state and bound oxygen, they can't release the oxygen that they have. So the net effect is the availability of oxygen in tissue can be reduced, and this can lead to hypoxemia. Another aspect of regarding the interaction of hemoglobin with nitric oxide is that in environments such as a tumor where you have low oxygen tension, nitric oxide can bind to deoxyhemoglobin in a way that's essentially reversible, but it doesn't lead to its degradation. And in effect, deoxyhemoglobin becomes a carrier of nitric oxide, allowing it to diffuse to other areas in tissue where it can be released and then effectively counteract the effects of hypoxia. A second aspect regarding nitric oxide and its effect on metabolism is that nitric oxide competitively binds to cytochrome oxidase. Now, cytochrome oxidase is the principal enzyme in mitochondria that accounts for why it's necessary for us to breathe oxygen. So we breathe oxygen, it gets bound to hemoglobin, transported throughout our body, and then reacts with this one enzyme in mitochondria, cytochrome oxidase, that reacts with oxygen to form ATP. So it's the principal vehicle for ATP production, aerobic metabolism in the body. And by inhibiting this, by inhibiting this reaction, you can have really two effects. So the idea is that you can look at nitric oxide binding to cytochrome oxidase as a kind of a braking impact. So you have a certain amount of oxygen that's being delivered to tissue. In the presence of nitric oxide, you can have vasodilation, and maybe that's more oxygen than you might need. So by binding to cytochrome oxidase, you can throttle back the activity of cytochrome oxidase. Now, it's possible that you can have too much of a good thing. So as you start inhibiting cytochrome oxidase, you can drive back electron transport to result in the production of something called superoxide. It is really an oxygen with an extra electron, and it allows you to then, which is highly reactive, and this can be very damaging to tissue. The second aspect regarding nitric oxide impacting on cytochrome oxidase is that as you reverse electron transport, you can then have pyruvate being inhibited from coming into mitochondria. That leads to lactate reduction in the cytosol, and this is what happens in tumors. So you have oxygen present, but the tissue can't use it. That's the Warburg effect, which is a major factor contributing to the metabolism of tumors. Okay. All right, so here I focus on some of the aspects regarding the synthesis of nitric oxide. And we know that there's really three isoenzyme forms of nitric oxide synthesis, whose reaction I show here, that are responsible for the myriad of physiological actions of nitric oxide. So here we see that arginine, which is an amino acid, can be converted to citrulline and result in the production of nitric oxide. Now, key here is to recognize that this reaction depends on oxygen itself. So here's one more example where the metabolism of nitric oxide and oxygen are very closely coupled to each other. In fact, you can think of it as a balancing mechanism, one versus the other. I also show here where we have the actual reaction involving nitric oxide synthase and how it affects the vascular endothelium. So you have arginine reacting with nitric oxide synthase. It generates nitric oxide. So it's not nitric oxide per se that produces the vasodilation, but it's reaction with an enzyme in the vascular smooth muscle called guanylyl cyclase. So guanylyl cyclase is the enzyme that converts GTP to cyclic GMP. Cyclic GMP is a mediator of a whole range of reactions in the body that can cause phosphorylation of enzymes and so on. And having produced cyclic GMP, this can then trigger a calcium-dependent reaction, which causes vasodilation. Okay, so here in this slide here, I briefly touch on some of the factors that cause the degradation of nitric oxide in the body. And I mentioned already the idea that superoxide can be reduced relative with nitric oxide, and it reacts with that. So you get nitric oxide with superoxide, producing this very reactive compound known as peroxynitrite. Now this can be very damaging to tissue, so you would imagine that there ought to be mechanisms that allow for a feedback control on this to modulate peroxynitrite levels. Turns out that one of the key mechanisms is, in fact, it reacts with nitric oxide and superoxide itself. So again, you have these small molecules that are being produced as a consequence of production of nitric oxide, and you constantly have these feedback loops, whether it's nitric oxide feedbacking with oxygen itself or with respect to some of its metabolites, controlling the levels of these very reactive compounds. Here I show an example that illustrates the impact that oxygen has on nitric oxide levels. So if you do a very simple experiment where you have in a petri dish, so you can start with just nitric oxide dissolved in some buffer, and nitric oxide can react with oxygen in the air, although it does so very weakly. So the red curve shows the time dependence of the change in nitric oxide levels when exposed to atmospheric oxygen. However, if you now add cells to this, you get a very rapid degradation of nitric oxide levels. Now, evidence that this degradation is in fact strongly dependent on oxygen is shown as this tracing here. So the red curve is in the degradation of nitric oxide with cells, but with 1% oxygen. The blue curve shows what happens when you have atmospheric concentrations, 21%. So it's strongly dependent on the concentration of oxygen. So what this tells us is there's this strong dependence suggests that the half-life of nitric oxide itself will be dependent on the concentration of oxygen. So in our lungs, where the concentration is close to atmospheric levels, the half-life is rather short because it can undergo a reaction with oxyhemoglobin to be degraded. As you go to other tissue compartments, and particularly in mitochondria, where the concentration of oxygen is going to be much lower, its half-life is proportionally much longer as well. So if you have, you recall I mentioned that nitric oxide moves about the cell by diffusion. It doesn't require a carrier. What that suggests is that the distance in which nitric oxide can travel itself will be strongly dependent on the presence of oxygen. So again, here we see that at higher oxygen concentrations that can occur like in the lungs, the distance that nitric oxide can travel is rather short. However, if we now go to lower oxygen tensions, such as occur in mitochondria, it has a much longer distance that it can travel. So this dependence of nitric oxide level on how far it can migrate and what concentration it may have when it undergoes this migration indicates that this impact on cells and the metabolism of cells will be dependent on these factors. So we know that nitric oxide, its actions on cell targets depend on the half-life of nitric oxide as well as on its diffusional distance. Now, here I just briefly highlight this. It's a little complicated slide, so I'll just walk you through this. So the different compartments of the cell have different oxygen levels, right? So depending on what that oxygen level is, you can have different values of nitric oxide. So here we have the endothelial nitric oxide form, which has a very high affinity for oxygen and can produce nitric oxide. Here's the inducible form of nitric oxide that modulates the immune response. So you get different concentrations of nitric oxide. Now these can have different impacts on key cellular targets. So this is soluble guanylyl cyclase. And so the level of very low concentration of oxygen, you can produce nitric oxide in sufficient quantities that this enzyme is maximally activated. Well, on the other end of the spectrum is the tumor suppressor gene, P53. So the ability for different targets in the cell to be sensitive to nitric oxide level is very much dependent on what the concentration of nitric oxide is, which in turn is modulated by the oxygen tension. Now, as you modulate the activity of these gateways to the various signaling pathways, you're going to have a large impact on the phenotype of the cell. So, for example, we know at high concentrations of nitric oxide, this can be cytotoxic, and it's the reason how the immune response combats infection. So you get a local infection, you get invasion of macrophages in that region. They produce huge levels of nitric oxide, some cases a 1,000-fold increase over what normally might be present, and it can just degrade the bacteria. In other cases, you have the idea, what's known as the epithelial-mesenchymal transition. This is the mechanism whereby tumor cells can be released from their sites of origin, travel about the body, and cause metastasis. All right? So here you have a number of key phenotypes that are very important in normal cell function as well as the propagation of disease. So these phenotypes depend on how the targeting aspects of the signaling agents are affected, which then depends on nitric oxide and then oxygen itself. So what it suggests is that disease outcome, whether it's going to be positively influenced or negatively by the presence of nitric oxide, and that's shown as an example here. So here we have just some cells grown up in various concentrations of nitric oxide. We can expose them to, say, 24 hours of nitric oxide, and in one case, you can get a proliferative response. If you increase the duration longer, this response is largely abated, so it's cytostatic. But with higher exposure times, you can get a cytotoxic effect. Okay, so what this slide here is meant to summarize is just bring us back to our high school teachings of chemistry, and it answers the question, how is it possible that you could get all these myriad of reactions given such a small molecule that can cause all this reaction in the body? And the simplest of all considerations is this is just acknowledging the valence states of nitrogen, all right? So these are all the different valence states of nitrogen, and in fact, it can react in a wide range of ways, producing either very stable compounds, such as a primary amine, or something as stable as nitrogen gas itself, or successively more reactive compounds. So this is nitrous oxide, which is an anesthetic, talking about nitric oxide itself. And these are the different forms of nitrite and nitrate, which when hydrogenated, you will form strong acids. Okay, so coming back full fold here, it's really ironic that Alfred Nobel, who made his fortune on focusing on how to stabilize nitroglycerin, his brother was killed handling this early on. So that motivated him to come up with a way to stabilize nitroglycerin, which led to the invention of dynamite, which led to his fortune, and hence the Nobel Prize funding, that the chemical structure that he was focused on here and really is quite similar to any of these other very small structures. It turns out as far back as 1847, they knew that nitroglycerin was vasoactive. Okay, they didn't know exactly why, but they certainly knew it was. And so here we fast forward 100 years later and find out that compounds that are substantially similar and pharmacologically identical, in fact, that's nitric oxide. So I think it's rather remarkable that the simple chemistry here has such a profound impact 150 years ago and now led to the Nobel Prize. Okay. All right, so I mentioned previously that when you look at the history of discovery and so on, we look at DNA. Okay, lots of things we learn from DNA, but the follow-on to the understandings of DNA and cell macromolecular protein production and so on has been all these omic technologies that produce tens of thousands of new jobs, new ways to treat disease, perhaps new ways of coming up with medicines. The alternative to this would be, what is it about the nitric oxide story that you might expect would have a similar follow-on effect? So is there some new class of technology that we might expect could follow on, given what we've already learned about the nitric oxide effect? And that's what I want to turn to now. Okay, so our focus is we're basically a technology development team. You might think, what does that have to do with nitric oxide? You shall see. So our focus has been to look at the use of near-infrared spectroscopy. Now this is, you've all probably done this experiment when you're a child. You put a flashlight to the back of your hand, you can see red light coming out the other side. You stick it in your mouth, your cheeks glow red, right? Okay. So in clinical medicine, that was known maybe 50 years ago, that simple kind of experiment allows you to monitor features of the hemoglobin signal. And here I show the optical spectrum. So this is in the near-infrared region. And it turns out that the spectrum of deoxyhemoglobin is notably different from oxyhemoglobin. You do at least a 2-wavelength measure, and you can quantify the levels of hemoglobin, its oxygenation levels, and so on. Okay, so early on when I came here at Downstate, I appreciated that, in fact, this phenomenon actually is quite interesting. Turns out we went on to figure out how to treat the measured intensity of light that emits from tissue and to actually reconstruct a 3-dimensional image. So we took teachings that was at the time known from X-ray CT imaging and applied it to this diffuse problem, which at the time was considered physically unsolvable. Turned out that the conventional wisdom at the time was wrong. We were able to come up with a simple equation which showed us how to do this, and it launched a whole new field and a new industry. So when we're using a key function of our use of this technology today, it's a neuroimaging technique. So I have an array of optical sources and detectors on the head. I can do a multi-distance measure. Here's the source. I measure increasing distances from the source. I get these banana-shaped pathways similar to the straight line paths in X-ray CT. And from this, you can reconstruct 3-dimensional images and get a whole time series of this. People are interested in this because as you think, you get a coupled hemodynamic response to the brain, to that added bioelectric behavior, which is nitric oxide mediated. And this phenomenon is measurable by fMRI, known as the bold signal. It's also measurable by FNIRS. Okay. So here I just simply show some of the early technology that we developed. This was our first tomography system, the size of a good-sized refrigerator. With time, it got smaller and smaller. Here we have this idea that you can measure 2 subjects simultaneously where they're doing a common task. With added time, we were able to miniaturize this technology further and further. So today, FNIRS technology, and we're not the only development team in this field, but FNIRS technology is widely accepted to be suitable for all subjects, from infants to aged adults, usable in all environments, even under the water, high-performance aircraft, space station, what have you. and can be used to explore all forms of human behavior, and it's being done so currently. Okay, so again, our focus here has been to introduce investigative resources, both hardware and software, that allow for new understandings regarding disease impact on tissue vascular behaviors. And this current applications, principally it's used as a neural imaging technique, studying learning, memory, language, sensory, social interactions among subjects, what have you. Here at Downstate, my focus has really been to apply this technology to look at breast cancer, okay? And so in particular, breast cancer diagnosis and more recently, treatment monitoring. It's also understood that the technology likely can be used to more broadly understand chronic inflammatory states across all disease processes. Okay, so here I'm going to focus now on our efforts in using NIRS technology to study breast cancer. So what we know from early pathology studies is that there's some key phenotypes that are common to tumors. And one of these is the idea of enhanced angiogenesis, which tumors, because they're growing, they say, I need more blood supply. They can induce the synthesis of blood vessels, the growth of blood vessels in the local tumor. But these vessels that are produced are not normal. So here we have an electron micrograph. This is a graph showing healthy tissue, where you have a very nicely fenestrated network of blood vessels. However, this is the equivalent result when you have a growing tumor. The vessels are enlarged, they're distended, you have dead ends, they're leaky, and so on. So the perfusion that occurs in tumor blood vessels is much more disorganized and less efficient than when you have in healthy tissue. And as a consequence, deoxyhemoglobin levels typically are higher. You have increased total hemoglobin because you have a higher number of blood vessels, and oxygen saturation levels are reduced. So this is the general understanding regarding the phenotype of enhanced angiogenesis. If you speak to a clinician who actually studies women for looking at the clinical breast exam, where they look to palpate the breast, right? So what they're looking for is lumps and bumps, okay? So our idea was, okay, how about building a device that can essentially duplicate what the physician hand's doing? So let's build a robotic type hand, and in this case it has 8 fingers to it. We can make 2 of these devices. They apply to the breast, and we can simultaneously articulate the breast in controlled ways while we're actually doing a time series optical measure. Okay, so this is the device that we actually created. It looks a little complicated, and it distinctively looks medieval, if you will. That's what it is. We called it the medieval freaky device in our lab, but turns out it worked out very well. And so again, what we're able to do here is a typical experiment would be, we could bring in 4 fingers from the side, so we can do a medial-lateral compression. We could have 4 fingers from the top. We can do a craniocaudal compression. And the amount of force that we apply, the duration, the timing, and all this is completely adjustable. And while this is happening, we're doing a time series optical measure. So the protocol we looked at was, okay, let's have the woman can sit comfortably in a chair, bring the device to the breast, right? We can now acquire a baseline measure. The woman's just sitting there comfortably resting. And then we go to proceed with these various types of precise articulations. And before we decided to really study the details of these articulations, because the analysis of this kind of data could get rather complicated, we said, well, why don't we do the easy experiment first? Let's just take a look at the resting state and see if there's something obvious in that may lead us as to understand how we might derive biomarkers for breast cancer. And when we did this, we came up with a very unexpected finding. And so here, what I show here on the left-hand side is, so if you just collect a time series from the breast and do a simple mathematical transform called a Fourier transform, which just looks at the frequency structure of the hemoglobin signal. So you expect a cardiac signal because the heart's beating. You expect a respiratory signal because you're breathing, right? Turns out there's a low frequency component as well, which is nitric oxide mediated, which is this pulsations of the blood vessels, okay? So if you're dealing with a healthy subject and you get recordings from one breast and you subtract that from the other, you might expect that they would be rather similar, so they would cancel, which is exactly what we see. When we looked at women with breast cancer, and it's overwhelmingly a unilateral disease, what we saw unexpectedly was this very large peak here, which we knew was associated with vasomotion. So we weren't expecting this at all. We went to the literature, said, well, this is too obvious not to have been studied in some way. Turns out a group from SUNY Stony Brook had studied it about 8 years earlier, but they used a technique called dynamic thermography, which is just looking at the heat signatures. So as you upregulate angiogenesis from tumors, you're going to have more blood flow, more heat in the tissue, and they could look at the time-dependent behavior in the heat signature. And when we compared their spectrum that they calculated to ours, they were almost superimposable. So with respect to what they showed, they then isolated the tumor tissue from women with cancer, did a xenotransplantation in animals. So now you can grow the tumor in that subject's tumor in the animal, expose the tumor to an inhibitor of nitric oxide synthase, and they completely abolished the behavior. So from my view, they nailed it. It's nitric oxide that's driving this effect, okay? But we're actually, given our technology, we can do imaging. So we said, well, why don't we take a look at the images of this? So we can get an image, three-dimensional image of this elevated time-varying behavior inside the breast. And when we did that, this is what we see here, shown here. So here, this is a woman with a right breast tumor, which is this one here. Here's the left breast. These are just different cross-sections through a volumetric image. And what we see is, rather than having contrast features that were specifically tied to the tumor itself, which is what you would get in MR, which is what you would get in mammography, no, the entire breast lit up. Now, why would that happen? Well, nitric oxide is a highly diffusible gas. It can bind to hemoglobin, especially under conditions where you have low oxygen tension. It can transport elsewhere in the tissue, be released, and cause a vasodilatory effect. So we were convinced that, based on this observation and what we could gather from the group's work at Stony Brook, this is overwhelmingly a nitric oxide effect. Right? So what this taught us is, okay, we're seeing something that's very obvious. And in fact, the diagnostic performance of this was we had AUC values over 90%, in some cases 95%. Okay, so this is a very promising measure. So the next question is, well, how do you systematically explore this? And this prompted us to do some rather basic thinking as to how one should go forward. And if you will, there are times when the right question to ask is, are you asking the right question? So in this case, with the question, what we wanted to do, explore was, how does one go about investigating dynamic behavior of any system? Okay, so and generally you have three options. One is a mathematical approach where you can have some kind of mathematical model that says, here's a set of observations, here's my model, and I can predict certain behaviors that I cannot directly observe. And this is commonly done, okay? It's done with fMRI Bold technique and so on. The next one is called a feature-driven approach, is what we started with the first time up. So we said, we have this complex time-varying behavior. Let's look at the Fourier spectrum, or let's look at this feature or that feature. And you can do that, and you can learn something from it. But it's not a particularly systematic approach. The third approach is something we all learned in high school. We learned it from our physics teacher, which is the idea to explore bulk properties of tissue. And by that I mean the thermodynamics of a system. Now that might sound like a little scary term or something, but when we think of thermodynamics, what we appreciate is the idea of entropy. Now entropy is a measure of randomness and a measure of disorder. So you can have a random system with very high levels of entropy. You can have a structured system with low levels of entropy. Now if something is structured, typically that means it follows a set of rules. It just didn't happen by chance. So an architect follows a blueprint to build a house, right? But typically the rules that he applies to build the house are the same rules that can be slightly modified to build an entire city or another city or what have you. So the concept that rule-based systems can be used to generate infinitely dense feature spaces has really been seen in many different contexts. And again, thinking back to our high school years, the question is, who did we learn this from in the first place? I'll give you a hint. It wasn't your physics teacher. Turns out it's your language teacher. So either your English teacher or your foreign language teacher was the first to introduce you in some systematic way to the understanding that simple sets of rules can generate very complex behaviors, in this case, all the different forms of sentences in the English or any other language. Okay? But when we think about languages, what we learn is that there's not just one class of languages. There are many classes of languages. Okay, so we have many forms of natural spoken and written languages, but there are computer languages, there are biological languages, the transcription of DNA, RNA, and so on. These all follow specified sets of rules, And these rules can be distilled down to rules of grammar and so on. So there's an alphabet with DNA and RNA, we know that, the triple codon sequences and so on. So the rules of linguistics have been applied to a wide range of fields. And this includes music, dance, animal song, cryptography, geography, and essentially any form of complex dynamic behavior can ultimately be represented as a rule-based system from which you can generate rules of grammar and so on. So we were motivated by this and say, okay, how do we apply the fundamental understandings of linguistics to explore the hemoglobin signal? And what we know in general is there are people who have been studying this for a long time. There's a fellow named Noam Chomsky, who back in the 1950s, as a linguist, developed these rules. Okay, these days it's known as the Chomsky hierarchy, and basically it's a set of rules that go from very simple to increasingly more complex, and so on. And when you examine these rules here, so I'll just give you a quick example. So we consider what's called a regular language, all right? So here, this could be something as simple as C-spot run. I think we've heard that sentence before, right? Or permutations on that could be run spot C, C run spot, C run C spot, okay? Now, all these permutations don't require that you remember anything about the sentence structure or the words that were used, okay? So that would be for a regular language. However, if you do have to remember that, you're now dealing with something which is called a context-free language, so you need to remember one word. So the sentence C spot C run would be an example where you had to remember something beforehand, which takes it out from being a simple regular language and makes it a context-free language. Okay, so when you explore this and apply it to different problem areas, it can be shown that in music, you have very simple scores in music, you have very complicated scores in music, so they cross different degrees of complexity in the Chomsky hierarchy. Same thing is true for animal vocalizations as well as written languages. Okay, so our hypothesis here is that hemodynamics is fundamentally a language. And having understood that, our goal has been to identify elements of language that speak to defining an alphabet, words, and sentences. So how do you do that? So this is a little bit complicated, so I'll just walk you through this. So there are two principal forms of hemoglobin that we can measure, oxy and deoxyhemoglobin, okay? So oxyhemoglobin is shown on this vertical axis, deoxyhemoglobin is shown on this horizontal axis. So if you just focus on these two axes here, you get 4 quadrants, right? So in this quadrant here, the values of oxy and deoxyhemoglobin are all positive. Here they're both negative. This one here, a deoxy would be negative, oxy would be positive, and vice versa here. So what we understood was there are different forms of hemoglobin that have been shown to be biologically relevant. For example, hemoglobin oxygen saturation. That certainly is relevant clinically. We also know that you can get a measure on total hemoglobin, right, which is just the sum of oxy and deoxy. Changes in total hemoglobin equal changes in blood volume, okay? So certainly changes in blood volume in tissue are clinically relevant. Turns out there's a third parameter that you can compute, which is just the difference in oxy and deoxyhemoglobin. So we have 2 independent measures and 3 dependent measures. Turns out you can come up with this 5-axis coordinate system that allows you to recognize all these compounds simultaneously. And when you do this, what you can realize is that in any one of these 10 sectors here, what differentiates one sector from another is the algebraic sign of deoxyhemoglobin or the exchange from, rate or the total hemoglobin and so on. So you have the 5 different components of hemoglobin depending on where I am in this state space here. I will determine what the amplitude, whether it's positive or negative. We can use this as a way to define an alphabet. So we have our 10 different hemoglobin states. The capital letters refer to values of hemoglobin components that are positive with respect to the temporal mean. Those lowercase letters are ones that are negative with respect to the temporal mean. And so we can come up with our language here is we have 10 different letters, positive and negative forms of the hemoglobin signal, and 10 different words. That's our language, very simple language. And the sentences that we can produce then correspond to sequences of these words, okay? So in terms of the states, so depicted here we have a series of, so here you can pick up a given point in time and just follow this point in time, or as it migrates in this state space trajectory. So when we do this, you get some sequence here. And from this, you can then determine that, well, I have the opportunity, if I'm in this state, and the next time point I'm in this state, I can ask the question, but how often does this happen relative to other kinds of transitions? When you walk through this, you can convince yourself that there's 4 fundamental parameters that are accessible here. So we can come up with a probability, which is how often you move from one state transition to another. We can do dwell times. We can actually compute 2 quantities that speak to how much hemoglobin is undergoing a transition from one state to another, and something related to that we call a flux. Okay, so where we're going with this is that we've come up with a discretized representation motivated by principles of linguistics that allow us to represent this time-varying property of the hemoglobin signal in the context of a fully defined language. And given these components of language, we can then look to quantify these features here. And now let me show you an example of what happens when you actually do that. Okay. So what we've done here is, so we have some representative information spaces that we have here. So one idea is the idea of a transition probability. So I have a certain state of hemoglobin that I can exist in at one point in time, and over time I can transition to other states. Turns out there's 100 such kinds of transitions you can do. We can quantify this, and what we observe here is that here, for example, in the upper right-hand corner would be transition from state 10 into state 1. This one here, lower right, left-hand corner would be from state 1 into state 10, and so on, and these are all the permutations. Without going on the details, what you can see is you have very high contrast here. So there is a preferred basis of hemoglobin to transition from one state to another, and the contrast here is over 300-fold. Now, when we look at this, so this is what we have for healthy. This is the tumor-bearing breast. This is an average of 18 subjects. Here is the difference between women with cancer and without, and here's the corresponding p-value. So these are the different kinds of transitions for which we see these changes. We can look at these other components of the hemoglobin signal, and your attention is here showing that each one of these different maps here, in fact, are very different from another, telling you that there are different classes of information that's being captured here. So where this takes us is back to what we understand about DNA. So what do we learn about DNA? So you have this basic understanding of DNA, but having appreciated that, we went on to build all these new technologies that have the common idea that there's different classes of information, right? And that they're all independent of each other. So effectively, we're arguing that something similar exists here. So we have a basic understanding what the action of nitric oxide is able to do. We can apply that using defined technology, and then from this, we can come up with interpretations that give you these very dense feature spaces, similar to the different forms of genomics that you get, or proteomics, and so on. And given these dense feature spaces here, we can show that, in fact, the information that you get from one class of feature space versus another are very different from each other. So this is where we are today. So the expectation is, and we're showing that it's disease sensitive. So the NIRS technology can be applied to almost any tissue in the body. This class of measures then allows you to get independent measures of dense feature spaces, similar to what they use with the genomic information. The big difference is, whereas genomic information doesn't tell you anything about the physiology of the body, this does directly. We know what nitric oxide is doing. We know what it means when you have increased or decreased levels of oxyhemoglobin and total hemoglobin. So for the first time, we feel that we're now having the opportunity to get dense functional measures on the body, essentially anywhere for any subject in any environmental situation, and can characterize this kind of information to give us detailed understandings of what the impact of disease is. Okay, here I simply show that we've moved away from the medieval freaky device, okay? So what we've now done here is we understand that there's something called smart textiles. So when you look to have some kind of set of electronic devices, sensors, and so on, we can introduce this into a fabric domain. So now your regular clothes become your actual sensors. And this is actually a picture of what we've developed. So we now have something that's a little bit more patient-friendly, right? So it's a brassiere containing these stretchable printed circuit boards that have our optical sensors and so on embedded in them. And this is a way we look to be able to do these kinds of studies moving forward. So to just summarize, okay, so we have experience in developing hardware and software for a wide range of capabilities. We determined that the resting state measures are sensitive to disturbances in nitric oxide levels. We've derived a formalism that employs a linguistic motivated model that reveals a broad and independent feature space. And these methods appear well suited to examine the impact that treatment responses, such as neoadjuvant chemotherapy, impact of chronic disease as mediated through various aspects of sustained inflammation. And I invite you to look at our website. There's some literature out there that if you're interested in learning a little bit more about this technology. And with that, I'll try to answer questions. Thank you.
Hi, Randy. Hi. I know you've applied this to more than the breast, and so I was just wondering if you might want to comment on some of the other applications.
Okay. So again, here it's Downstate. I'm actually trained as a cancer biologist. That's why I gravitated in this direction. But having applied it to studies of the breast, and other people have, the tool or the application space for people who are most interested is studies of the brain. So if you will, pathology of the breast, of course, is an important clinical problem, but the basic physiology doesn't quite have the pizzazz as the physiology of the brain, okay? Because there's lots of things we'd like to know about how the brain works under all sorts of conditions and so on. So today, the NIRS technology is continuing to advance. We're getting higher density systems, much more lightweight and so on. And what's been demonstrated is, so if I'm doing an EEG measure, which is a bioelectric measure of brain function, and I move my shoulders or move my head or something, the electrical signal coming from the muscles will just overwhelm the signal from the brain. It's about 100-fold higher the amplitude. Hemodynamic measures turn out to be much less sensitive to that. So we have people doing jumping jacks. We have people jumping out of airplanes. We have Navy SEALs working in the sea and so on. So the NIRS technology can be implemented in such a way that it's very resistant to motion artifact. So you can get a fairly good quality measure of what's happening inside the head itself, and then you couple this to well-defined protocols and so on, and there's a great deal that you can learn from this. And it's being used. In fact, a little over a year ago, there was a group in Germany that showed for the first time that you can actually read people's thoughts. So these are people with ALS. This is the Lou Gehrig's disease. So that's what Stephen Hawking had, the British astrophysicist who recently passed away. So in the limit, these subjects, they can move nothing. They can't even blink their eyes. So how do you communicate with them? So ALS is principally disease of the peripheral nervous system. Central nervous system is intact. They can hear, they can see, they can think. How do you communicate with them? So using technology that we developed, the group at the University of Tübingen went on to show that they can train subjects, basically ask them yes or no questions, so to which they have a high confidence they know the answer. So, the capital of Paris is Moscow. No, that's not the capital of Paris, or capital of France. So you can come up with protocols that you can convince yourself that the answers you're getting or the signals you're getting from the brain do correspond with a degree of reliability to a yes answer or to a no answer. They published this in PLOS Bio a little over a year ago, showing that, in fact, they were able to communicate with subjects who are completely locked in. So that's one example of an application where it has been able to overcome a challenge that previously was not possible. And so it takes time to develop new technologies and applications. Experience shows it's at least 20 years until after you have something that's really feasible before you can go on to something to do something that's really useful clinically. I'd say in the near space we're about 10 years into that process, so we still have more time to develop and so on. Any other questions? So I'd just like to comment, so I'm new to the nitric oxide field. I had no expectation of necessarily getting involved in this. I was drawn into this because of the results that we got from our breast cancer, where we saw that the entire breast lit up. And I said, how is this even possible? And it was through not only what we had seen in the literature, but when you drill down hard and look at the basic biology of nitric oxide, the results that we saw are actually entirely predictable. Okay, and this idea so we have a way of measuring an inflammatory response in the body basically anywhere. Well, that's not actually easily accomplished. You can do PET techniques to do this and inject radioisotopes and so on, but outside of that, there's really not a radiologic tool that allows you to objectively quantify the magnitude and the time dependence of an inflammatory response. And measures of inflammation are really important because, if you will, every chronic disease results in a sustained inflammatory response, in particular, most of your psychiatric diseases. So what we now think, forms of perhaps Alzheimer's, but certainly various forms of schizophrenia, your mood disorders and so on. The expectation is there's an underlying inflammatory response that then has negative impacts on synaptic profiles in the brain, and that can impact behavior. And we now think that using this rather simple tool, we have the opportunity to quantify these measures, not only in the breast, but elsewhere. Yes, Richard?
From an old clinician, what comes first, the inflammation or the chronic disease?
Okay. Well, you might be more positioned to answer that question, but at least from my understanding with terms of cancer, it's now considered that the existence of a sustained inflammatory response is a prerequisite to cancer. So you do not get cancer unless you first have the sustained inflammatory response. And again, when you think of the inflammatory response, that's the inducible form of nitric oxide, nitric oxide synthase that is triggering that. And again, the amplitude of that response can be 1,000-fold over what you normally get with the modulation of the endothelial cells lining your blood vessels. Yeah. Thank you for that question. Any other questions? So, well, thank you very much. And there's some literature out there if you're interested that this speaks to. Oh, am I missing? Oh, yes, please. Thank you.
Thank you. If you're using NO to do all of these studies that you do, all right? It seems to me, imaginable, that there's unlimited amount of studies that can be done.
I'm taking hard. Can you just hold?
I said, it seems to me that would be an unlimited amount of kinds of studies that you can develop using NO.
Well, okay, so we know that nitric oxide essentially affects. Yeah, thank you. We know that nitric oxide essentially affects all parts of our body, right? So your vascular space is everywhere, right? And so in terms of the different forms of nitric oxides, there are biases. Some of it's mainly in our endothelial cells, others in nervous tissue, and so on. Okay, in any case, so nitric oxide is everywhere in your body, and it's everywhere in biology, yes. So, yeah, I mean, when it comes to measures, so you're not going to be able to easily measure nitric oxide directly in the body, or at least not non-invasively. But we can measure its impact on the vascular response. We can measure that by the fMRI Bold technique. We can measure that by the optical method, and there are other tools that could be used to do that. So yes, I think if I'm answering your question properly, the range of reasonable experiments or meaningful experiments that you might be able to conduct is truly vast. Right. Well, thank you very much, then.